Hello. It is my pleasure to present on the Immunold NeXT platform for tumor immunogenomics and biomarker discovery. I am Christelle Johnson, and I am a field application scientist at Personalis. Personalis was founded out of Stanford in 2011 and is the leader in advanced genomics and analytics solutions for precision medicine and personalized therapies. We are located in Menlo Park, California. The lab is CAP accredited and CLIA certified. Our solutions span research to clinical applications, and we've built a robust infrastructure to support our assays and the amount of data that we produce. We have received samples from across the globe in support of major applications such as translational research and biomarker discovery for immuno-oncology, as well as neoantigen-based personalized cancer treatments. There has been tremendous progress in cancer therapies, specifically with immune checkpoint blockades with difficult to treat types of cancer, such as breast cancer. In 2019, we've witnessed the approval of the first immunotherapy in breast cancer, and this is a combination of PD-L1 checkpoint plus chemotherapy as first-line treatment for metastatic triple-negative breast cancer. If we look at the overall landscape of cancer therapies today, there are a lot of promising new treatment modalities for cancer. Most of these therapies are either FDA approved or currently in clinical trials as monotherapy or combinations targeting various steps in the cancer immunity cycle or based on the immune status of the tumor. Despite this wide spectrum of pathways and targets, the majority of patients receiving these therapies still do not derive benefit. There is a critical need to expand benefits to a larger population of cancer patients by implementing a biomarker-based approach. However, there's a lack of strong predictive biomarkers that take into account the genomics and immune component of a tumor. We also know that cancer biology and mechanisms of resistance to different therapies can be complex, and understanding these is key to identifying more meaningful biomarkers. The current biomarkers to predict patient response are unable to accurately determine those individuals that will derive benefit from checkpoint blockade therapy, such as those that block the PD-1 pathway. The two immunotherapy biomarkers currently in clinical practice are PD-L1 expression and microsatellite instability status. Yet, as shown here in this slide, these markers do not always enrich for responders. For example, as illustrated on the left, patients that may be PD-L1 negative may still respond to treatment, while patients who are PD-L1 positive may not respond. The utility of these biomarkers also varies by cancer type. As the landscape of predictive biomarkers continues to evolve, it is clear that PD-L1 expression and microsatellite instability status are no longer sufficient on their own in identifying patient populations that are more likely to benefit from immunotherapy. We see multiple studies that begin expanding beyond these two metrics and into other parameters such as tumor mutational burden, gene expression signatures, germline and somatic HLA status, functionality of the antigen processing machinery, different cancer pathways, tumor-immune status, TCR clonality, and others, with the goal of identifying biomarkers with stronger predictive value. In order to profile the genomics and immune component of tumors, there is a need for a high-content assay that provides visibility to all of these elements and enable this comprehensive assessment. In addition, as we move from conventional treatment modalities to more personalized precision approaches, the information required to guide these treatments needs to be more and more comprehensive to capture the true biology. So we begin to see a shift also from smaller scale panels that mainly focus on cancer pathways and identifying mutations in the tumor that can be targeted, to broad whole exome and transcriptome assays looking at all 20,000 human genes to account for multiple biological systems. This has been the focus at Personalis. Advances in technology has enabled us to engineer clinical-grade comprehensive assays, whether it's tissue-based or plasma-based, to better understand the tumor and immune interplay. Our flagship product, Immunold NeXT, is developed to enable comprehensive characterization of the tumor genomics, but also the immune component and tumor microenvironment from a single tumor sample and a paired normal sample. We maintain a sample-sparing approach and can work with 4 FFPE slides to derive DNA and RNA, which we then interrogate for multiple biomarkers. We see the oncology landscape evolve beyond a single biomarker approach, and this is exactly what this high-content and scalable assay provides: identification of the biomarkers of today, listed here and beyond. By interrogating DNA and RNA features from the same sample and all 20,000 human genes, we can begin to combine multiple biomarkers and produce more meaningful composite outputs with increased predictive power. This is a unique capability of the platform and has allowed us to identify a composite neoantigen score, which I will talk about in more details when I present a clinical case study utilizing this platform. But this is becoming the trend in the translational space in an effort to better understand mechanisms of resistance and identifying patients who are more likely to respond to different types of therapies and drug combinations. In order to make such an approach practical, we innovated at multiple steps in the process to overcome the current challenges in the field today. The clients we work with and who have implemented Immunold NeXT as a biomarker strategy into their clinical trials have struggled in the past with having to parse out a limited and precious tumor sample to send for different specialized testing. These are often done with different technologies and with multiple vendors. So it extends time to analysis and complicates integration of the data from multiple sources. At Personalis, we have created a more efficient process. From sample receipt, we follow a dual extraction protocol and unique library creation, and process the sample on our Immunold NeXT platform to produce data-rich analytics and multiple key biomarkers. I do want to address here the sequencing technology we use at Personalis. As you might know, sequencing is not a trivial process. And to give you a flavor here of some of the challenges in conventional sequencing, the blue plot shown in the example above the gene here is a gene sequenced with a standard exome kit at high depth. It is clear that there are regions that are insufficiently covered and others that are completely devoid of coverage. These gaps and inconsistencies naturally impact the quality of the data. What we've done at Personalis, we went on and systematically identified where these gaps reside and designed probes to target and substantially augment these areas to achieve the green plot and uniformity in coverage across the exome. We do this supplementation for all 20,000 genes with Immunold NeXT. We also published on this technology, and as shown in the graph on the right side of the slide, Personalis technology, illustrated with the green line on the top here of the graph, achieves high sensitivity and clinical grade performance compared to other standard exome kits listed in the figure. This technology is at the core of all our assays. Also, unlike a traditional exome and transcriptome assay, the design of Immunold NeXT includes unique features. In addition to the augmentation of all 20,000 genes, we sequence at increased depth for the exome and transcriptome. We also added a unique boost of clinically actionable cancer genes at 1000x, a boost of immuno-oncology regions such as the TCR and BCR genes from the transcriptome. We've improved the coverage of HLA genes, we placed probes in microsatellite regions, and targeted oncoviruses as well to create a comprehensive assay for immunogenomics profiling. We co-developed analytical solutions to make sense of the data produced by Immunold NeXT. and report on multiple biomarkers shown here. We continue also to make advancements at the computational level. We developed DNA and RNA pipelines that we mine for various outputs. We built our own internal tool for HLA-related features, whether it is typing from germline or somatic calls and HLA genes and loss of heterozygosity detection. We developed a neoantigen prediction pipeline trained on immunopeptidomics data, where we also integrate binding and presentation parameters for neoantigens, as well as other analytical capabilities to generate a comprehensive data set and support reverse translational studies from clinical samples for the identification of the next generation of biomarkers. In addition to Immunold NeXT for translational studies and biomarker discovery, the assay enables clinical reporting through a diagnostic test called NeXT DX, based on the 1000x boost of actionable genes that we included in the design. With NeXT DX, we generate a diagnostic report that goes back to the physician. The report provides a summary of somatic alterations identified in the tumor, interpretation of these variants, whether there is an FDA-approved therapy associated with them, and whether there are US-based matched clinical trials that the patient could benefit from. We also provide 2 biomarker outputs, an exome scale true TMB output, and we report on microsatellite instability status. Another assay I'd like to highlight here is NeXT Liquid Biopsy. This is a non-invasive method to identify novel biomarkers. We have taken a different approach than the traditional panel-based liquid biopsy assays and have developed an exome scale assay, sequencing at high depth, and also including a significant boost of clinical genes. Keeping with the comprehensive theme for all our assays, we wanted to provide the full picture of the solid tumor through immunogenomics profiling with Immunold NeXT, but in addition, we now have an exome-wide look of the plasma as well. Some of the key applications we're looking to enable from the plasma are cell-free tumor mutational burden, assessing tumor heterogeneity, acquired resistance mechanisms, clonal evolution, expand to neoantigen detection and tracking, but also continue at different time points to study tumor dynamics. We believe the plasma assessment is complementary to the tissue biopsy in capturing the true biology of a tumor. In the next few slides, I will walk you through a clinical case study to demonstrate how Immunold NeXT can be utilized to understand potential mechanisms of resistance and how we were able to derive some insight on responders and non-responders by using this high-content assay. This study is conducted in collaboration with Dr. Sequan Zhang from the Innova Shar Cancer Institute. To describe the cohort, we obtained samples from patients with late-stage unresectable melanoma who have received anti-PD-1 therapy. We looked at the first 52 patients in this set for whom we had clinical outcome results. Here, clinical response was determined based on RECIST criteria, based on best overall response and progression-free survival. Responders included both partial and complete response, and non-responders included progressive disease. The 2 groups were matched in terms of demographics, age, gender, and other factors. Then we took the baseline tumor biopsy sample and the paired normal sample, sequenced and analyzed these in our lab, and we explored readouts from Immunold NeXT analytics and plotted several metrics to identify mechanisms of resistance and response profiles. We first looked at TMB as a key biomarker in melanoma. We believe that true TMB is optimally calculated from the exome, and we report on SNVs and indels. We observed slightly higher TMB in responders than in non-responders, but with overlap between the groups, suggesting that in this particular cohort, TMB does not seem to be a strong predictor of response and did not show strong association with progression-free survival. Since TMB is a surrogate biomarker for immunogenicity, We also computed neoantigen burden as a next step. When we calculated neoantigen burden in this cohort, we found that it is more strongly associated with response and improved progression-free survival with more significant p-values. But we still observed some unique profiles that we were interested in exploring further through immunogenomic characterization with the platform. And I will highlight these in the next few slides. The first example is a partial responder, but with high neoantigen burden. We looked at multiple parameters to explain this profile and found alterations at the HLA level that might play a critical role. Each of the HLA class 1 genes, A, B, and C in this partial responder had two distinct alleles from typing the germline. However, by looking at the tumor, we found that there was a chromosomal deletion resulting in the removal of one allele in all three of the A, B, and C genes. This HLA LOH event is potentially detrimental to the number of neoantigens that can be presented for immune surveillance in this particular individual and may explain partial response to checkpoint blockade. We were interested in looking at the frequency of HLA LOH events across different cancer types. So we plotted a few cancer indications from our database of samples that have been profiled with Immunold NeXT. Using our HLA LOH detection tool, we observed a wide distribution of these events across various cancer types. Notably here, we recapitulated what has been observed independently by another key study with non-small cell lung cancer, where HLA LOH frequency is close to 40%. We also see breast cancer falling in the mid-range of the spectrum with regards to these events. Now, going back to our melanoma case study, we were curious about another patient in the non-responder group, but who had relatively high neoantigen burden. In this patient, we detected somatic mutations impacting two HLA class 1 genes, in HLA-A and V. One is a stop gain mutation and one is a splice site mutation, respectively. Both mutations had a high allelic fraction and high variant effect impact, suggesting that they may be affecting the function of the HLA molecules. In addition, we predicted which HLA the patient's neoantigens were likely to bind to, and in fact, 40% were predicted to bind to the two mutated HLA alleles. Therefore, although these neoantigens were present at high frequency, they failed to be effectively presented by the HLA. This could be a potential mechanism of immune evasion and possible explanation of lack of response in this case. In another patient, a non-responder, we observed a somatic frameshift variant in a large proportion of the reads within the Beta-2 Microglobulin gene, which is a key component of the antigen-presenting complex. This frameshift variant potentially limits correct HLA class 1 folding and antigen presentation for the majority of class 1 peptide MHC complexes. This is consistent with other studies in the literature showing similar mechanisms of resistance. Now, if we look at the neoantigen burden again and take into account alterations impacting HLA genes and the antigen-presenting machinery, we can produce an adjusted neoantigen burden as a composite biomarker. And by doing so, we observe tighter correlation with clinical outcome and better separation of the groups here. This is just one example of what we enable with the platform and the various analyses and insights that can help identify novel and more meaningful biomarkers for different cancer types. We also explored other outputs from the assay, and here we looked at the TCR clonality in the subset of cases. We observed significantly higher clonality and number of TCR beta clones in responders when compared to the non-responder group. This initial data suggests that a more clonal profile at baseline may be predictive of patient response to anti-PD-1 therapy. This represents, again, one example relating to the immune component within the tumor microenvironment and enabled by Immunold NeXT. We see TCR clonality being used as an emerging biomarker in the field that also shows a diverse distribution across cancer types. We use TCR clonality in conjunction with other biomarkers to come up with more context-specific or tissue-specific composite outputs. Finally, this case study demonstrates what is possible with the platform, the various analyses and insight that can help produce more meaningful biomarkers from different cancer types. What truly makes this platform unique is that it provides our customers with the ability to simultaneously and comprehensively characterize both the tumor and immune-related components of the tumor microenvironment from a single sample, and study the complex and dynamic interplay between various cancer types and cell types within the microenvironment, with the ultimate goal of improving clinical outcomes to various immunotherapies and designing better strategies for difficult to treat cancers. Thank you for listening to this presentation.